Good morning. Welcome back for the third in this series of lectures by Professor William Dialek from Princeton University and the Graduate Center. His title today you see on the board, Statistical Mechanics for Real Biological Networks. And let me remind you that while this is a talk for a more sophisticated audience, there is a general public talk this afternoon at 5:30 across the hall. I hope all of you will come to that. It should be a very interesting talk. And with that, I give you Bill.
Thanks, John. So yeah, I decided that it might be useful to do this, at least in part, with slides, because there are a few places where I think it's important to see not just the theorist's conception of the data, but actually real data. So what I'm going to talk about is this fact that a lot of the things that happen in biological systems are the result of the interactions of large numbers of components. And there are examples which are extraordinarily familiar and sort of obvious, like the flocking of birds or the schooling of fish, where the phenomena that catch our eye obviously result from the interaction of many elements. You can have a flock of thousands of birds. But more subtly, even if you ask about the structure of a protein, well, the protein is held together by the interactions among large numbers of amino acids. We know, for example, that if you want to think of the elements of the network as being the amino acids, the states that they take on, or the different possible choices of amino acid at that site in the polymer. Evolution is sort of exploring this space of possible sequences, and we know that many different arrangements of these microscopic states, that is to say many different amino acid sequences, in fact give rise to the same protein structure. This example is from a family of proteins called the serine proteases, which are molecules that, well, for example, live in our gut and cut other proteins as we digest them. And the details of the chemistry hinge on this particular set of amino acids being arranged in 3 dimensions just right. It turns out that there are serine proteases that are found in bacteria and serine proteases found in us, where if you look at the amino acid sequence and you try to line them up, you see almost no similarity. Of course, since they're called serine proteases, there is actually a serine in both cases, but that's about it. On the other hand, it was therefore a tremendous surprise when people solved the structure of these proteins by X-ray crystallography, and you could take the 2 molecules and put them on top of each other, and the entire molecule was the sort of trace, the backbone, was the same within a few tenths of an angstrom. And these 3 residues that are critical for the chemistry were exactly on top of each other, plus or minus 0.1 angstrom. So same structure means recognizable fold of the backbone, and in the case where you know exactly which items are relevant for the chemistry, the precision is much higher. So you can think of protein structures as being held together by the interactions among large numbers of amino acids, and the dynamics of evolution as exploring this space of amino acids. We talked yesterday about the events in early embryonic development in flies, and what I'd like to emphasize here, which perhaps I didn't emphasize fully yesterday, is that really cells are deciding their fate in the course of development based on the expression levels of these many genes, and these many genes are interacting with one another. The dynamics here is the dynamics of proteins interacting with DNA and reading out the information. And then, of course, there's the case of neurons in our brain. This is an image from the laboratory of my colleague Michael Berry at Princeton. This is already a generation past, but it starts to give you the idea. This is a piece of the retina from a salamander, and the green circles are the bodies of cells in the salamander retina, and they send their axons out, eventually forming these bundles, which ultimately come together to form the optic nerve. And the black things that you see are electrodes. And the point of this image is to emphasize that technology is now to the point where we can record the activity of almost all of the neurons in a small neighborhood, and as you'll see in a larger neighborhood. And it's reasonable to think that almost everything that the brain does is not the result of the action of single neurons, but rather some more collective effect that results from the activity of large numbers of neurons. And so presumably when we imagine something or recall a memory or make a plan to move our arm, it's not one neuron doing something, it's a whole population of them working together. And so the notion that here what matters is some sort of collective behavior of the network as a whole is something that I think is intuitively appealing, but once you think about it, here the idea that there's something collective going on is sort of immediately apparent when you look. Here I think you need to think a little harder, you know a few more facts, and here you really need to think quite a bit harder. You're sort of inverting the usual view. Normally you might think that the amino acid sequence is fixed and the protein is moving around. Here I want you to think about the fact that as you go across evolutionary time, the sequence is evolving. But since these proteins have a particular function in the cell, to some extent, you're evolving and exploring the space of sequences while maintaining this three-dimensional structure and the corresponding function. And so each of these networks, the network of amino acids, the network of genes, the network of neurons, the network of birds, there's a local variable that you can keep track of. There's something that reflects the state of the entire network. There's a dynamics for how you get from one state to the other. And one of the questions that you might ask about the system is, what is actually the set of states that are sampled in real life? Okay? And in the case of neurons, the set of states that you're sampling has to do with your capacity for conveying information. The structure of that state space has to do with different intuitive ideas about how information is represented. In the case of networks of gene expression, it really corresponds to how many different kinds of cells you can make. There's also, again, sort of more textured questions about the shape of this distribution over states, which has to do with whether things break up into modules or whether there's really something collective, and so on. So you can ask, I think, parallel questions in all of these examples. And I should say that I asked our friends here to do this on that half of the board so that if questions come up, I can use this half, not being entirely comfortable without the chalk in my hand. So please feel free to interrupt. So in the physics community, there's been a long-standing hope that these sorts of effects, where large numbers of elements come together to do something collective, could be described by statistical mechanics. So in the context of a flock of birds or a school of fish, the idea that all the birds agreeing to fly in the same direction is something like all the spins in a magnet lining up is, I don't know who was the first person to have that intuition, but it should be a very long time ago indeed. Okay? In the context of neurons, there's a long history, but surely a milestone is John Hopfield's paper. Essentially, what happened in this paper is that, which launched a whole field, was he wrote down an approximate model for the dynamics of neurons, clearly throwing away lots of things that we know are true, but in proper physics tradition, trying to pull out the things that one thinks are essential and leave out the details. In this approximate model, there's a Lyapunov function for the dynamics. And so one could then think about the dynamics as being the network sort of rolling downhill on some energy surface. And that, not only that, but you could understand then that the local minima of the energy surface, where the dynamics would stop, would be the answer to your computational problem. So you'd imagine the dynamics of this network, it starts somewhere and it moves along, and where it finally ends, that's the answer to your question. It might be the memory you were trying to recall. It might be some other computation you were trying to do. And so there's a mapping between this sort of physical intuition of the energy landscape and the computations that the network manages to do in a collective way. And this work was done, as many of you know, just as the tools, the mathematical tools for describing the statistical mechanics of systems with disorder were emerging, and in the context of spin glasses. And there were lots of analogies, counting the number of energy minima in these kinds of models and counting the number of energy minima in corresponding models of spin glasses were sort of very, very closely related problems. And this led to a very fruitful interaction between the fields, which is sort of ensconced now. If you look at the physics ePrint archive, you'll notice that the category for the statistical mechanics of disordered systems is actually disordered systems and neural networks, which I think is a kind of remarkable flow of things from biology back into physics. One realizes somehow this is one big field, right? If it's really true that the things you're looking at are collective and you can describe them using statistical mechanics, one of the things we know is that in statistical mechanics, if I have a very large system, then the parameter space of that system, the natural things I can vary, like temperature, for instance. If I make a multidimensional parameter space, that parameter space breaks up into phases. And in the limit that the system is infinitely large, the boundaries between the phases are sharp. And so it becomes natural to ask if what we're looking at, if we're describing the collective behavior of a biological system and we're using ideas from statistical mechanics, well, what's the phase diagram and where is the real system in this phase diagram? And as many of you know, there is an idea which I guess in its original form arose not too far from here. I think Per Bach was at Brookhaven when the first papers about self-organized criticality were written. And so there was a set of ideas that perhaps biological systems, or maybe particularly in Per's vivid imagination, systems, non-equilibrium systems more generally, somehow could organize themselves to critical surfaces in their phase diagram. And this was marvelously provocative and not terribly successful. And I think the reason is, and even, I mean, what John did was extraordinarily successful, but it's taken a very long time for it to have an impact on how we think about real neurons. And I think what's been difficult in this whole theme, even if you think about the seemingly more obvious case of all the birds agreeing to fly in the same direction being like all the spins in a magnet agreeing to point in the same direction. To a large extent, these are metaphors. And they're incredibly powerful metaphors. But in physics, we appreciate metaphor as well as the next guy, I suppose, but that's not a theory, right? You're supposed to be able to calculate something. And I would say, for instance, particularly in the ideas about criticality, I would say that they died a kind of whimpering death, not because there was something that you checked and it turned out to be wrong, but rather because the theoretical ideas were over here and people were doing experiments over here, and it was never clear what it was in the data that you would, what quantity would you compute from the data that would test the hypothesis that was being enunciated over here. Okay? So what I'm going to tell you today is a set of theoretical ideas which in some ways are completely unoriginal. Okay? I'm not going to tell you anything theoretically that one couldn't have said many, many years ago, and that were probably said by somebody in some context. Okay? What's new is that the experimental situation has changed dramatically, and I think there's a possibility of building very direct links between this body of theoretical ideas and the real experiments that people are doing today. And that's why I think it's an exciting time. I actually do that. And so this is all, we've been at this for a while now, and we've been thinking, we started by thinking about neurons. We then started thinking about proteins. We had a little excursion playing with the spelling of words just to illustrate things. We came back to neurons. We started thinking about proteins, flocks of birds, and so on. And all these papers, first of all, they're all on the archive, but they're also all accessible through my website. So, and we're still at it. So what I'm going to do is I'm going to explain to you something which many of you probably already know, so perhaps it's a review. The idea of the maximum entropy construction, which allows you to make a bridge between the things you can measure and models that have the flavor of statistical mechanics. And as I say, the thing that's going to be very important about this is, well, we'll unpack it a bit, but I think what I want to emphasize is that this is a tool for producing models that one recognizes as models in statistical mechanics, which nonetheless are completely grounded in experiment. That doesn't mean that they're correct, right? You still have to check.
Between maximum and minimum free energy. Statistical mechanics balances off energy versus entropy.
So the maximum entropy construction that I'm going to use is one in which we're building models of a probability distribution whose entropy is maximized subject to a certain set of constraints. If the only constraint was that the average energy come out to be the one that you measure, then that would be equivalent to minimizing the free energy. But these are systems in which there isn't obviously an energy or anything. So, let's say the distinction between maximizing entropy and minimizing free energy is the addition of a constraint, or can be thought of that way. I'll unpack this in a little bit. I'm going to spend a fair amount of time talking about the example of neurons in the retina, both because that's where we started and it's the one where, well, in some ways I think we've made the most progress. And I'm going to say a little bit about proteins and birds and so on. And then I'm going to end with a little perspective. So let me...
Where are the bees? Birds and bees, yeah.
Yes, no, I got the joke. I was trying to think of something clever to say in response.
Never mind.
Well, there are, of course, species in which sex is actually collective, but...
No longer in ours.
Right, yeah, well, yeah, anyhow. That's right, yeah. Here it's kind of, for humans, it's a sort of specialization. Okay, so... Actually, of course, the decision, for example, of insects to change hives, right, is another one of these collective, right? They all go, you know, if something goes wrong with the hive, they all start exploring, and they come back and kind of convince each other, okay, let's go this way. It is, in fact, another good example. The difference is that I actually know people who can track several thousand birds at once. I don't know anybody who can track several thousand bees at once. Smaller. Good. So why is this sitting here? It's sitting here to emphasize that now, I don't want to say that it's routine, but it is possible to do experiments in which, unlike the classical experiments, you know, most of what we've learned about the brain that involves actually knowing what the neurons are doing, and we've learned a lot about the brain without actually recording from neurons. For example, that, you know, if you get an injury in this part of your head, then it's this particular function that you lose, and so that tells you that that function is somehow localized in that part of your head. Most of what we learned about the brain by recording the electrical activity of neurons was learned by recording from them one at a time. And, you know, at various moments, people have made the obvious observation that with tens of billions of neurons in your head, why do you think you're going to learn anything at all by recording from them one at a time? So it's sort of remarkable that we've gotten anywhere, although, of course, we might just be fooling ourselves.
What about time average, space average ideas? So you can study population by studying time series of one.
So, absolutely, one of the reasons why we have gotten somewhere is because one has looked very carefully at the dynamics of what the individual neurons are doing over time and their relationship, for example, to sensory stimuli in the outside world and so on. You could have imagined a brain in which the relationship over time between what one neuron is doing and, let's say, what's going on in the sensory world is so convoluted by virtue of their interactions with each other that you still couldn't decipher it. But it would appear that that's not at least how some parts of the brain work. But so there's been a big push over the years to record from more and more neurons at the same time. And there are different technological approaches. In this one, the idea is that you have the advantage that the retina is more or less flat, and the neurons are projecting out, if you will, the bottom of it. So you take it out of the animal and you lay it down on an array of electrodes. And if the spacing between the electrodes is comparable to the spacing between the neurons, then it's plausible that you'll pick up the signals from all of them. Okay. Now, you actually still have to sort them out, because if you have a neuron sitting here, probably its electrical signal will show up at several electrodes in the neighborhood. And so there is a signal processing problem that you have to solve here, which is quite challenging. But my colleagues seem to have solved it. And so they are now at the stage where they're recording using devices like this from roughly, let's say, 85 to 90% of the neurons in a neighborhood, and that total number is 180 or so. So it's a neighborhood with 200 plus, and you're getting about 80 to 90% of them. And that's an interesting regime to be in, because in this part of the retina, if you look at one neuron and you see how far out can it reach and touch other neurons, and you draw a circle, there's about 200 neurons in that circle. So this is kind of the group of interacting cells. And if you sort of went over one, there'd be another group of interacting cells. So we're approaching the point where you can see the entire network. And this is the network of cells that has responsibility for representing some small part of the visual world. Okay? And so these experiments are exciting in a number of ways. I mean, one of them is to realize that everything that the animal knows about that little part of the visual world is passing, you're picking up those signals. And so if you want to think about the problem of processing visual information, you now have the opportunity to ask that question in a much more direct way than you ever could before. This is all of the visual information you get about that little piece of the world. You could try making sense out of it. Okay? See whether you can do as well as the brain can, even as well as the brain of a little salamander. So let's do a little mathematics. Yes. Somehow I skipped past this one. This is important. So what is it that we actually want to know here? Right? A really good question would be, how do you get from one state to the other? That is to say, can you make a model of the dynamics? And I'm actually not going to talk about that. It's not because I believe it's not interesting. I'm just not going to talk about it. Another question you might ask is, what's the distribution over states that you actually sample in real life? And I tried to argue that knowing something about this distribution tells you something interesting in each of these cases. In a sense, looking at this in the context of the retina is telling you about the vocabulary with which the retina is speaking to the rest of the brain. You can ask about its capacity, you can ask about its structure, and so on. There's also a question, of course, in the specific context of the retina, or neurons in general, what do these patterns of activity mean about what's going on in the outside world? And I'll say a few words about that, but I think before one answers that, you actually just have to characterize the vocabulary. So to make an obvious point, you can't answer these questions directly by experiment. And the reason is that the state space is too big. Okay? So you can't go out and measure this probability distribution. Right? If I have n neurons And in each little window of time, they either generate an action potential or they don't. Then there are 2 to the n states. And for reasonable n, 2 to the n is larger in natural units than the lifetime of a graduate student, and therefore you can't measure this. More importantly, it's larger in natural units than the lifetime of the piece of tissue sitting in your dish. Okay. So you can't measure all the states. But what you can do, of course, is to measure various averages and correlations and so forth. So you can take your data and compute something from it that has the form of an average over the duration of your experiment, and plausibly, again, assuming that things are statistically stationary and they're reasonably ergodic and so on, those should be the expectation values drawn out of this distribution. So the question you might ask is, if your experimentalist friends tell you that there's some set of these that they can measure reliably, what can you say about this probability distribution? And the answer, of course, is, in some rigorous sense, nothing. Because if you measure any finite set of these that doesn't actually determine 2 to the n things, then there's an infinite number of these distributions that are consistent with it. But there is one that's special, and that is the distribution that reproduces what your experimentalist friends tell you, but otherwise has no extra structure. So it's the minimally structured distribution. What does minimally structured mean? It means that the states that you're drawing out of this distribution are, in a sense, as random as possible while still being consistent with these things. And that means that this distribution has the largest possible entropy that's consistent with these measurements. Okay? And this is going to get us a direct connection to ideas in statistical mechanics. So now, yes. Sorry, let me be clear. If you pick this set, then the maximum entropy construction is unique. Of course, you could have picked a different set of things to match. And I don't think there's a unique, so there certainly isn't a unique choice. I'm not even sure we know much about whether there's a best choice. To make it clear, you have built a model which absolutely agrees with certain experiments. And the other thing you've done is the opposite of what theorists usually do, which is you've taken all of your theoretical prejudices out, right? You've said, I don't want to put any structure in there that I don't have to put in order to match what the experimentalists tell me.
And that's unique.
That's unique. Now, the 2 observations are, first of all, you did put in your theoretical prejudices when you decided which things to match. And the other one is, there's absolutely no reason why this should agree with this should be a good model of the real system, right? So just because you happen to match some set of experimental quantities and otherwise don't have any extra structure, it could be that the quantities you matched don't really capture the essence of what's going on, and so the model you build will fail completely. Or fail a little bit, and then you have to think. Fail completely is in some sense fine. It's the one where it fails a little bit that you have to think hard.
Are there any cases where you say, well, we measure this, and we make this maximum entropy model, and we measure a second thing, and we put the 2 together?
So,
Relation is?
in fact, the natural way to talk about this is to say that there's a bunch of things you could measure. And what your experimentalist friends tell you is this set of expectation values. And then if I want to construct the maximum entropy distribution, it's a straightforward calculation to show that the form of the distribution is this. And this distribution, I'm going to say more about it in a moment. So some of you already know this. What this is saying is that the distribution is an exponential form. In the exponential, there's one term for each of the expectation values that you tried to match, and it has a coefficient.
So
And
these measurements are completely independent of each other.
they don't need to. Sorry. Let's be careful what we mean. So I decide that I have these 2 quantities I want to match. It may or may not be true that that's true, but that doesn't matter, right? So I don't need, you might suspect that if all of these quantities are actually correlated with each other, then matching all of them isn't really adding much relative to matching a small set of them. But there's no, right? There's no constraint. You're allowed. I mean, the expectation values are all measurements on the system.
So you say the system does this, and
Yeah, the system does this.
you get one of them right. Well,
Then you might get the other one right for free because they're very correlated,
Right.
or you might not.
yeah. Okay.
Okay, and in fact that's part of the whole spirit, right? You're going to take some finite set of these that reflects some combination of your intuition about what's important and the limitations of the experiment, right? So what you shouldn't do is pick one of these where there's a gigantic error bar, right? Don't do that. Okay, you can, but then you're off in a world of problems of statistical inference that I would rather. I mean, it's an interesting world, but it's a different one.
So how many measurements do you have to make?
Hang on.
I'm trying to figure out have you tested the idea by taking some statistical problem, we actually know the answer.
Absolutely, yeah. No, this is a yeah. What I would say is that everything is well behaved. That is to say, there don't appear to be any surprises where it takes an anomalously large amount of data to become reliable or anything like that. So we'll get back to this in just a second.
To get back to this question of the minimization of something like a Gibbs free energy versus the maximization of the entropy. So if G would be U minus ds, right? So in some sense, it gets back to the question of these coupling constants, which would naturally enter into the internal energy, but you're not really considering that here. But you seem to be considering.
So let me, for the moment. So I sort of have to oscillate back and forth, right? There's a well-defined mathematical problem for which I never need to say words like free energy or anything like that, right? I have a probability distribution. There's a function of that probability distribution I could compute called the entropy. There's another function of that probability distribution I could compute, which is the expectation value of F1. I mean, it's a fairly simple function, right? I just add up F1 times P, and I can F2 and so on. I want you to find the maximum of the function entropy subject to the constraint that these things all equal their experimental values. That's a math problem. Don't say the word free energy. Okay? Of course, in a moment, so the point is that is a perfectly well-posed mathematical problem, and this is the answer.
So
Except the G's have to be tuned so that you get back these things correctly. And you'll notice there's exactly as many of them as there were constraints. And that's a hard problem, by the way, in general. Yes?
Are these quantities edited in some sense? Yes.
Pick anything you want. These could have different units.
But why exponing them?
Oh, because that's the solution. So the problem we're solving is that, let's just describe these as states S, right? I have the entropy, and then I want to maximize this entropy, but I want to constrain that the certain expectation values come out right. So that means that I want to be sure that if I sum F1, which is a function of S, PS, it comes out to equal F1 from experiment. And so I do that with a Lagrange multiplier. And then I do the same thing for G2, and so on. And now, if you just take this whole quantity and do that, you'll discover but that's the solution. Now, the F's are additive in the exponential as a consequence of this. I mean, it's additive only in the sense that when you have multiple constraints, the terms in the Lagrange multipliers add up. And for example, as I mentioned, the F's could in principle have different units, in which case the G's have different units. They're not additive in the sense that they live in the same space or right, whatever you want.
Can I ask you a more philosophical question? So you have essentially k data points.
Well, I have, right,
And
yes.
you have in model that has k parameters.
Yes.
And as somebody who works with data and models, it's if you have two data points, if you have 2 points, right?
Oh.
Straight line through.
Of course.
You never know whether the straight line is the 2 points are actually.
Yes. So let me make two observations. First of all, the statement that you have k data points should be unpacked a little bit. You have k constraints. These expectation values that your experimentalist friends measure, they presumably measured by making many measurements, right? In particular, if they're determining k functions of the state of the system, they had better have made many more than k measurements, right? So in fact, underneath all this, there's a very large amount of data, which has been distilled down to k quantities. You're then going to match you're correct that what you're doing is matching those quantities exactly. And then you have to ask, does this actually describe the system? So in some sense, you could say that you're doing the equivalent of fitting a line to 2 points. Of course, those 2 points are essentially exact, right? Because they're based on a very large quantity of data. And so now you have to, given that you only have 2 points, in some sense, the only thing, and you want to match them exactly, the only thing you can do is draw a line through them. Anything else would be making stuff up. And then you can ask, well, does the real system fall on a line? And so it is very much analogous to that question. Well, I'd like to think it's a little more sophisticated than that, but in some way it's not, right? You're taking certain aspects of the experiment literally, and you're asking, is that enough to explain what else is going on?
What are the correlations you're talking about?
Well, an example of it's perfectly possible that F1 is sigma 23, sigma 47. And so its expectation value is a correlation function. So it could be anything. OK? So now I want you to bring back your physics intuition, having banished it for a moment. Let's bring it back. You remember that if you have a physical system and there's one thing you know, which is the energy, and one way of thinking about thermal equilibrium then is that the equilibrium state is the state of the system which is as random as possible, has maximum entropy, but the average energy is what it is. Okay? And in that view, which is a little bit inverse to the usual view, this quantity beta, which appears here, is just like the g's before. It was just the Lagrange multiplier that imposed that the average energy come out right. Now, we know that it has a name. It's called the temperature. And we tend to think of that as having physical significance. But I don't know. You can sort of trade between the temperature and the average energy. You know that. So that's what's going on here. And you realize then that to finish the construction, you have to do the inverse of statistical mechanics. So the usual thing in statistical mechanics is I would tell you the temperature, you would compute the energy, or its derivative, the specific heat. Here, I have to do it the other way. Somebody has told me the average energy, and I have to go tune the temperature until it comes out right. Except I don't just have the average energy, I've got lots of these things, and so I have to tune them all right. OK. The other piece of physics intuition is that if you notice the form of the distribution, it is the Boltzmann distribution with that energy. And it's really important that this is now an exact mapping. This is not life is like a river and birds are like magnets, right? It's the same equation, right? So that means that if you have you've chosen some set of expectation values to match, then you have built a model which assigns to every state an energy. And the prescription of the model is that the distribution is the equilibrium distribution with that energy function. Okay? This doesn't mean that the dynamics correspond to rolling downhill on that energy function or doing Brownian motion on it or anything like that. It just means that the distribution corresponds to the Boltzmann distribution of that state. So let's think about what some of these things are in the context of neurons. Some of the expectation values that I might try to match are things like just the average probability that each neuron is spiking or silent. So that's the expectation value of the sigma i's. And if I do that, then the energy function is just a bunch of spins in a magnetic field. in magnetic fields, plural, because the magnetizations, that is to say, the probabilities of spiking from the different neurons could be different. I could also decide that I care about the pairwise correlations. So I want to be sure that I match not only the average probability that this neuron fires an action potential, I want to match the joint probabilities that pairs of them fire action potentials in the same small window of time. Then in that case, the energy function is a collection of Ising spins in magnetic fields interacting in a pairwise fashion. This case, of course, I didn't write it down, but this case, of course, you can do everything analytically. This case, you can't, because in general, these interactions will be as complicated as, well, there's no reason for them to have a simple form, because the correlations have a complicated form. There's another thing you could try and match. Instead of just matching the probability of spiking versus silence for single cells, you could match the probability that m out of the n cells spike together. And then in that case, the energy function just has this is just a global potential acting on the magnetization. And actually, this one we can also do analytically. That is to say, right, by analytically, I mean that there's nothing hard about doing the inverse problem. You can do it. You can write the formulas out.
For some set of stimuli, or
so
what's going on?
Right. So what's going to go on in the experiment is it is the equivalent of taking a walk in the woods. But these are salamanders. Well, the retina is no longer attached to the salamander, so it's not taking a walk. And salamanders don't live in the woods. They live larval salamanders live in the water, and they're amphibians, remember. And so what you do is you take movies of murky underwater scenes in a reasonably natural setting where you sort of stir things up and things are flowing past, and you show that movie to the retina. And it's statistically stationary in the sense that it's a very long movie, and if you compute anything sort of, well, the first 10 minutes, of course, the retina is getting adjusted to stuff. But then, if you take the next 10 minutes and the 10 minutes near the end of the experiment, any quantity you compute had better be the same, or else you know that the retina was dying. Okay? There's no drift in the movie, so if there's drift, it's in the dish. Okay? And again, we could ask things about the relationship of the patterns of activity to what's actually going on in the visual world, and I'll say a couple of words about that, but mostly not. I'm just going to talk about sort of the set of states taken on by the network, and what does that look like. Okay? So let's do this one. And since in these experiments they're recording from 180 neurons or something like this, I can take groups of n neurons out of this neighborhood and study them and solve this problem. And so, as I say, if I just take the data, I can construct this potential, which, of course, I should have plotted for you, but for some reason I didn't. And I can do that sort of even in the same experiment. I can do it again and again and find out whether there are error bars. So, in what sense are different groups of n neurons similar to one another? Is there some typical network whose statistical structure I'm getting at, or is everything mired in details? You'll notice, of course, that what this is saying is that the energy in the mapping statistical mechanics depends only on how many neurons are firing at the same time. Okay? That means, of course, that there are many ways of having the same energy. In fact, there are n choose m ways of having m neurons fire at once, so you can calculate that factor. And so that means that I can compute the entropy of all the states that are associated with a particular value of the energy. Furthermore, if any of our analogies to statistical mechanics are correct, you expect that both energy and entropy are extensive quantities. So as the group of neurons that I look at gets bigger and bigger, they should both be proportional to the number of neurons. So let me plot the entropy per neuron versus the energy per neuron. And let me do this by going through this construction in groups of 20, 40, 80, 160 neurons from this neighborhood. And in fact, I can even think about extrapolating to infinity, right, by plotting this versus 1 over n and seeing if there's something well behaved. And what you see is that if you take groups of 20 neurons, you know, there's an error bar, but it's not huge. And by the time you're looking at groups of 80 neurons, these are really very, very small. And the if I take different groups of n neurons, the standard deviation of the energy per neuron associated with having half a bit of entropy per neuron is this big. It's not very large. Okay. And by the time we're talking, I mean, that's when we're only talking about 20 neurons. By the time we're talking about 80 neurons, you can't see the error bars anymore. And so,
how do you actually measure this energy?
We measure the probability that m neurons are firing at once, and the maximum entropy distribution that's consistent with that has this form. And so I can, so if I ask you what's the probability, so the maximum entropy distribution over the states has the form of this, okay? If I now ask, what is the probability that this is true, then it's just some combinatorial factor, which depends on m and n, times e to the minus v of m. And so since I know this, since I can compute this combinatorial factor, I can determine what this is. But of course, this combinatorial factor is, up to taking a log, the entropy associated with that particular energy.
How will you relate this to the operation of this this set of neurons, this associative memory? And this
Hang on. Okay, we'll get there. So far, this is just a warm-up exercise. Okay, so better get moving on. Okay, so yes.
Is there a plateau at the high end?
Well, in the data, there might be. That gets less obvious as you look at larger and larger systems. And we don't really know, right? Remember that as I go out this way, I'm talking about sampling states which are less and less probable. And although this is 1, then you think, well, e to the minus 1 isn't a very big number. But this is per neuron. So e to the minus 160 is a small number. So I don't know whether this is here or there. Okay, I mean, it's real data, right? So the surprising thing is that if you go through this exercise, the answer is that within error bars, the entropy is just equal to the energy. Now, let me emphasize for you that this is really weird. Of course, this model is incredibly simple. And so you could just say, it's weird, go on to the next thing. But let me explain to you why it's weird, because the reason it's weird is really interesting, and that turns out to capture something which I think is quite important and survives as we try to go to more realistic models. So let me remind you that the reason one plots the entropy versus the energy is because that plot contains everything you want to know about thermodynamics, right? So if I try to compute the partition function for a system, I do this. But of course, since the only thing that appears in the Boltzmann distribution is the energy, you don't really want to sum over states. You first want to sum over all the states that have the same energy, and that's just a number. And then you do the sum over. So I could think of this, call it n of e, where n of e is the density of states. But you could also think that in the limit that there are many, many states, this becomes almost continuous. And so it's really e to the entropy associated with that energy. And so the integral that you're doing in order to compute the partition function is just that. And since both the entropy and the energy are extensive, there's really a factor of n of the size of the system sitting in the exponential. And so that means that you expect that the integrand will be dominated by the saddle point. And the saddle point, of course, is that. which is the definition of temperature. And the way you can tell, for example, whether you're approaching a critical point is that the second derivative will start to get small, right? And that's the point where, if you work it through, the specific heat is starting to diverge and so on. So everything you want to know about the thermodynamics is all encoded in S of E. Okay? If S is actually equal to E, then something very strange is going on. First of all, we're having this discussion essentially at temperature equal to 1, right? There is no temperature, so it's just a matter of units. So, and if the entropy is equal to the energy, then in fact there is no saddle point, or every point is a saddle point, or however you want. You're at a sort of, or alternatively, not only does the second derivative of the entropy vanish at a critical point, but every energy is a critical point. So there's something really weird going on here. Okay? So hold the thought. So what we really want to do is not this Mickey Mouse problem where we remove all the labels on the neurons and just count how many of them are firing. We really want to keep track of the probability that each neuron fires. We want to keep track of the correlations between pairs of neurons. And we want to keep track of this overall probability that k things are firing at once. And so what I need to do is to take the measured expectation values of all of these quantities and do the inverse problem. And one can do that from about an hour's worth of data. This is a hard problem, but we now have control over it. So as an example, with 100 neurons, you might see this correlation function for the expectation value of sigma I, sigma j. When you're finished doing the inverse problem, the j's end up looking like this. They're interestingly more uniformly distributed, but also sparser, which makes sense, right? Correlations extend farther than interactions. To convince you that we actually solved the inverse problem, we can do Monte Carlo of the model that we have, compute the correlation functions from the model, compare the correlation functions from the data, and measure the difference in units of the error bar on the measurement of the correlation that comes from the fact that there was only an hour's worth of data that went into this. Right? The little time slices are about 20 milliseconds, so we have hundreds of thousands of samples, but that still gives errors. And convincingly, we are reproducing things within an error bar. Of course, it could be that this is actually the wrong thing to do, because we very carefully reproduced the measurements. But the measurements, in fact, because we're matching correlation functions, and there are roughly n squared of them, and we have 100 neurons, n squared is 10 to the fourth, and did we really make many more than 10 to the fourth independent measurements? Just because we made however many measurements we made, we don't know whether they're all independent of each other. So you have to check that you haven't overfit, right? So in the technical sort of learning theory or machine learning language, you want to be sure that it's not the case that the correlations that you happened to observe are themselves spurious. And you fit them, and we fit them well, but right? Maybe you shouldn't have. So the way you check is you divide the data into parts, and you use some subset of the parts to learn these parameters, and then you check the log probability of the data in the rest of it and ask whether those come out to be the same, and they do. So we have not overfit, right? If there was something about what was going on in this part of the experiment that was true but coincidental, and you fit it, then when you got to this part of the experiment, it wouldn't fit, and that's not what you see. So we can solve the inverse problem, and we're not overfitting. This is not very interesting, but it's really important to be sure you can do it. So how do I convince myself that this model, again, it is definitely the model that has as little structure as possible while still reproducing these aspects of the data. But how do I know whether it's a good description of the real system or not? So I have to check something else other than what I used to construct it. So if I have a small network, like 10 neurons, I could just, 2 to the 10th is not large compared to the lifetime of the tissue. And so I can just measure the probability of each of the states, here presented as how often they appear in time, but it doesn't matter. And you can see that the observed probabilities and the probabilities that are coming out of the model are essentially equal to each other, except down here, where if they're occurring at 10 to the minus fourth per second, they occur once per hour, and so that's the experiment was an hour long, so of course there's a big error bar here. In contrast, you notice that correlations here are all very small. And so you might have thought that you could almost neglect them entirely. And in fact, that argument has been made, that when you record from neurons, you find that they're weakly correlated. And so maybe you should just pretend that they're all independent of each other. Even in the context of 10 neurons, were you to pretend that all neurons were independent of each other, if you predicted the probabilities of occurrence of the different patterns, you would be wildly off. You would get the general trend that having 1 neuron out of the 10 fire is more probable than having 2 neurons out of the 10 fire. But once 2 neurons out of the 10 are firing, your prediction is almost anti-correlated with what actually happens. So if you try to predict which 2. And I'll draw your attention to the fact that there are particular patterns that you would predict should happen once per year. And in fact, they happen a couple of times per minute. And so on. They could cherry pick other examples. Yes?
The eye is supposed to take a noisy input signal and map it onto a stored pattern like a
Well, a brain, yes.
Okay.
This is.
How is this giving, the old Hopfield idea?
Yeah. We're not there. We're not. Hang on, okay? I still have to show you. I mean, you're sort of granting me that somehow this all works, and I still have to convince you that it works, okay? Or at least I would like to take you through the reasons that we believe. I mean, the claim here is really quite startling, right? It's that these statistical mechanics models that people have been dreaming should describe the system actually describe the probability distribution of states accurately, okay? And I want to be sure that I have reason to believe that before I take anything else about the model seriously, okay? So the other thing you can do, or another thing you can do, is, for example, compute higher. So with 10 neurons, that's fine. With 100 neurons, that's not going to work. So let's do something like compute 3-point functions. And let's look at the 3-point function in the data versus 3-point functions in the model. Of course, there's 100 to the 3 of them, so there are lots. It turns out it really is important to have this business of matching not just the pairwise correlations, but also this probability that several things are firing at once. And now you see that things work. You'll notice that the absolute errors are falling. The larger the network you consider, the more accurate you are. And furthermore, the overall scale of this is very, very small indeed. So the scale of the data is maybe typically 10 to the minus 4th. The errors that you're making are more like 10 to the minus 7. This really is working extraordinarily well, okay? And again, you matched pairs, and you're checking triplets, right? So there's no reason why this has to work out.
Matching the pairwise correlations and the single correlations, and also something having to do with the.
There's just a global thing, which is the probability that m out of the n are on.
There's a parameter for that.
There's order n parameters, right? Because it could be that 0 fires, 1 fire, 2 fires, and so on. But there aren't n squared or n cubed. There's order n. So in fact, one of the reasons that we noticed that if you just did this, right? If you only kept if you only kept the pairwise in this, then the really interesting thing that you were getting wrong was you were starting to predict that the probability of the entire network being silent was off by a few percent. But it's silent a third of the time, half the time. So over an hour, if you get that wrong by a few percent, that's actually very significant. And we were sort of charmed by the idea that what we were getting wrong was the probability of silence. and so we went to fix precisely that. That is to say, we decided to fix the probability. I have to admit, we haven't checked. If we only fix the probability of silence, is that enough? What we decided to fix was the whole probability of m things firing together.
What time scale?
We're taking time and slicing it into 20 millisecond bins. And the reason we chose that is if you look at the correlation functions as a function of time difference, that's about the widths of the features. So that seem natural. Nothing that I'm saying depends very crucially on that number.
So, but you're assuming that there's a stationary distribution.
We assume there's a stationary distribution, and we discuss that. Well, we have, of course. I mean, in some sense, if these were actually in order in time, which we can do, then the statement that these look the same as those is also a test of stationarity.
Can we interpret that middle plot there? Does this say something about how long range these correlations
How
are?
long range the effective interactions are.
Looks like it's all over the place.
Exactly, and the reason is that neurons have arbors that reach out, and if you draw a circle, there are lots of neurons inside that arbor. In the case of the retina, it's of order 200, and that's thankfully how many neurons we're recording from. By the time you're in cortex, neurons have arbors which are a noticeable fraction of a millimeter, and if you take a cubic millimeter of the cortex, that has 100,000 neurons in it.
It's not your dad's Eisenhower.
Exactly, right. Well,
Well, I know what I'm talking about anyway.
yes. It's not, depends who your father was.
Which gives rise to the idea of using
Right. So yes, okay, so these are long range. You'll notice they come in both signs. A reasonable fraction of the triangles are frustrated. It's not a ferromagnet, okay? It's a spin glass. But it's also, by the way, just as for reference, if you took the distribution of these elements and made a correlation matrix at random by choosing out of that distribution, you would get, and then solve the inverse problem, you would get a network whose thermodynamics, if you will, is very similar to the real network. If you took the distribution of these elements and chose at random from that distribution, well, then you know what you get. That's the Sherrington-Kirkpatrick model. And that's not what this network is. Okay?
What's the physical meaning of plus and minus?
That the neurons tend to fire together or tend to exclude one another. So this is all by way of convincing you that, and of course there's more to do, right? I mean, I'm showing you a sampling. We're in the process of writing a very long and I must say somewhat tedious, but I think important paper convincing ourselves that, and hopefully at least one or two referees, that if not, readers, that this really works, okay? And as I say, we started here with, groups of 10 neurons 5 or 6 years ago, and the fact that things worked kind of touched off a great deal of interest in using this in other contexts and solving the inverse problem and so on. But what one's really waiting for, I mean, statistical mechanics is interesting and the n goes to infinity limit. We can discuss whether 200 is getting close to infinity, but 10 surely wasn't, okay? So all the exciting stuff, and in fact, the experimental program to try and record from 200 neurons at once was driven by this, okay? That we wanted to see if we could see the collective effects that we thought should be there. So if you believe that this is a reasonable model, we can now take it seriously as a statistical mechanics problem. So it's a big network. It's not infinitely large, but it's big. Where are we in the phase diagram? So there's a lot of things we could check, but let me make, oh, sorry. Before I do this, let me make one other observation. Because this is a spin glass, it means that the energy function has many local minima. Okay? These are, these are the stored memories of the Hopfield model, if you will. The question is, do they mean anything biologically? So if you play the same movie over again, and you come back to the same moment in the movie, you will discover that the state of the network is not exactly the same as it was the last time you played the movie. Now you might think, well, that's because it remembers. No, it's because there's a little noise. Okay? The states of the neurons fluctuate even in response to the same stimulus being repeated. What's remarkable is that, of course, this construction, which makes absolutely no reference to the visual stimulus, tells you that there's an energy surface with multiple minima. So that means that I can, for every state that I see, I can figure out where I am on this energy surface, and I can label which basin of attraction I'm in. If I play the movie over again, the system visits the same basin of attraction with probability almost 1, despite the fact that the microscopic state, exactly which neuron is firing, fluctuates from trial to trial. Yes?
Trial, you're probably going to learn something, so the distribution.
This is the retina.
Change, so there's no learning.
There's adaptation, but that's on a shorter time scale. So when you start showing the movie, if you had shown a movie with different statistics, the system would do something different. But once you've been showing a movie with the same statistics for 5 or 10 minutes, it's stationary, and it's not learning in the sense that the central brain learns. So now, where am I? So that was to emphasize that it's not only the case that the model works in the sense that it describes the probability distribution, it also works in the sense that there are structures that are implied by the model which seem to actually mean something for the retina in ways that had nothing to do with how we built them. Okay? So now, where am I in parameter space? Well, there's one direction in parameter space for which I have an intuition. So let me take that one, and we can discuss others, but this is the one that I think is most compelling. And that is that if I have a maximum entropy distribution of this form, and I take all of these parameters and I scale them up and down together, that's equivalent to changing the temperature. So let me look in that direction. And so what do I want to compute? Well, you know, it's thermodynamics, right? Why not compute the heat capacity? So if you were looking at, I think this low one is just the independent neurons. So if you sort of looked one neuron at a time and averaged, then the heat capacity would look like this. Okay? Kind of broad, featureless. 1 is the real temperature, is nothing special. As you start to look at groups of neurons that are larger, see 20, 40, and so on, you start to see a peak developing. And so remember that heat capacity, right, it's related to the energy, so it's also extensive. So let's divide by the number of neurons, so we get a specific heat. And now this is the picture. 1 neuron at a time is out here, 20 neurons, 40 neurons, 60 neurons, 80 neurons, 100 neurons. We can go further now. The first thing you notice is that it's actually growing super extensively at the real system. So the specific heat, if you will, of n neurons grows faster than n. And the reason is that as n is becoming large, the specific heat as a function of temperature has a very high peak, which is located within 1 over n of temperature equals 1. And so what this is a signature of course, is that the system is sitting on a critical surface in its parameter space. And then let me emphasize, this is not at all what we were looking for. Okay? In fact, everything I've told you is not what we were looking for. When we first did this, our presumption was that by building the maximum entropy model that was consistent with pairwise interactions, what we would do is to clean out all of those uninteresting effects of the pairwise interactions and reveal the higher order things that must be really important. And the surprise was that the model worked. And there wasn't this other stuff that we were looking for. And then you realize that, and then the next surprise was that you're actually, once you start taking the model seriously, you realize that something like this must be going on for reasons we could talk about. But anyhow, that it would be this clean, we weren't clear about. So because there was this failure of the idea of criticality that came from an overemphasis on ideology, I would like not to spend time in the last couple of minutes of the real part of the talk discussing why the system should be at a critical point, okay? Apparently it is. And what we'd like to do before declaring victory and claiming we've discovered the system operates at a critical point, we'd like to do some more direct experiment. So remember that the state of the network is not just a consequence of its anatomy, it's a consequence of its adaptation to the stimuli that you've been showing it. So maybe by showing it drastically different stimuli for a short period of time, we can knock it out of this state and watch it come back and show that being on the critical surface is something that's actively maintained. That would be a little more compelling than just noticing this, right? But let me emphasize, this really is quite a surprising thing to find.
For each end?
In fact, we fit many separate models because you can choose 20 out of the 106 out of 200 many times, and this is the average. There's also an interesting question, which is if you take the model for n equals 100 and integrate out 20 of the spins, do you get anything that looks like the model for 80? And the answer is yes, although of course not exactly, which is why the models are doing better at describing things when they're larger, right? That is to say, even if the system in the large is described exactly by this model. You'd expect that if you don't observe all of the elements and you integrate out the things you're not observing, you generate other effective interactions which you're not keeping track of.
Are you trying to build in any, you know, synaptic plasticity into this model?
So this is a description of the distribution as we see it, right? How it gets to be that way, whether it's adaptation or synaptic plasticity or whatever, we're not saying anything about. Let me go a little further. I want to spend a couple minutes telling you that you can do the same thing in other contexts. I mentioned the problem of families of proteins that have different amino acid sequences, and I pointed to the example of flocks of birds. So how do I do this for flocks of birds? Well, I could try to make a model of the entire probability distribution of the flock, their positions, their velocities. Let me do the simplest thing first. The simplest thing is I know where the birds are, and I know how fast they're moving. I just want to make a model of the distribution of directions in which they're flying. Okay? So every bird now has a unit vector which points in the direction of his flight. And I want to make a maximum entropy probability distribution that's consistent with that describes that. So what expectation value am I going to hold fixed? My intuition is that birds talk to their neighbors to decide which way to fly. So let me compute your average correlation with your neighbors, with your first shell of neighbors. And that's one number, and only one number. And then after that, everything else follows, right? If you can match that one number. And in fact, that's also not so hard to do because the flock is rather highly ordered. So for the cognoscenti in the audience, right, you're writing down a Heisenberg model, and since you're highly ordered, you're in the spin wave limit, so everything's fine. You can do everything. The only cheat that we do is we take the birds that live on the border to be given. Because, of course, they're different than the guys in here. They can see out. They presumably their directions are influenced not only by their neighbors, but by other things. And so to simplify our problem, we take the flight directions of the guys on the border to be fixed. And then we study, in a sense, the propagation of directional order through the flock. This is the typical nearest neighbor distance. And then the model, once we match the average correlations in here, then the rest of the correlation function is completely determined. And I hope you agree that the fit is pretty damn good. Furthermore, if you look at flocks of different sizes, you discover that this scale on which the correlations change sign, the sign change here is partly because of the way we normalize. We could go into the technicalities if you're interested. That scales with the size of the flock, right? Because there really isn't a correlation length. And if you decide that you want to compute some bizarre 4-point correlation function, you can get that right too, including this funny wiggle somewhere in the middle of the flock. Now, because this is a directional correlation, the fact that it's long-ranged is not surprising. That's Goldstone's theorem. Okay? However, if I showed you the correlation function for speed fluctuations, it would look almost the same. And there's no Goldstone, right? This is not, you're not breaking a symmetry for the magnitude of the vector, right? Fluctuations in the magnitude of the velocity vector also have long-range correlations. And that is a sign that this network is sitting at a critical point, okay? Which we have not analyzed yet, but this was, again, the first step, trying to convince ourselves that these models actually work. We're almost there with the speed fluctuations. In the context of families of proteins, this was done, there's a beautiful experiment where you sequence all of the antibody molecules that are made by a zebrafish, and you can do this for many different fish. And we've made maximum entropy models for the sequence diversity in a small region where the diversity is maximal, which is exactly where the antibody recognizes the antigenic challenge. And one way of showing you the success of the model is to plot probability versus rank, the famous ZIP plot. And we do this for the actual sequence ensemble that's observed and for what the model predicts. And you can see that they line up perfectly, except, of course, the model is a model of probability distributions, not of samples. And since they only have tens of thousands of independent samples, you eventually fall off from the curve. But the agreement is very good. If you pretended that all of the amino acid substitutions were independent of each other, it would be a disaster. And furthermore, this is all reproduced from fish to fish, except for one guy who's sick. Yeah, it's the immune system, after all. And this plot of probability versus rank, although it would take me a moment of algebra to show it to you, this is the plot of entropy versus energy, sort of turned around, right? Because this is log probability, which is an energy. Rank is how many states are there whose energy is less than this. And, well, its logarithm is the entropy, or almost the entropy. And so this plot is really entropy versus energy, and the straight line is, again, S is equal to E, which tells you that you're sitting at a critical point. So, sorry for going a little long. What I hope to have convinced you of is that this new era where we can do experiments on tracking 1,000 birds at once, recording from 100 neurons at once, sampling sequence ensembles with hundreds of thousands of sequences, means that using the maximum entropy idea, we have a path from real data to models that are the statistical mechanics models that our intellectual forebearers were hoping would describe these systems. Except that now, instead of pulling those models out of a hat or using them as a metaphor, the data actually determine not only that it's a model kind of like this, it is this particular model with this particular set of parameters. And quite remarkably, those models make quantitative predictions about all sorts of aspects of the data which turn out to be right, okay, which was by no means guaranteed. And once you sort of have that groundwork, you can say, all right, now let's take them seriously as a statistical mechanics problem. And the astonishing thing is that in these different contexts, it seems like all of these models are poised somewhere near their critical surfaces. And if that's really true, then I think we've discovered something important. And so I'm cautious to declare that we know that it's true. There are lots of hints, and they're coming in different directions. It could still happen that the data get better, and we become more careful, and everything narrowly misses the critical point. And in each system, it misses in a different way, and that's what's important. And the whole thing kind of falls apart. And that's not excluded yet, so I don't want to overstate where we are. But I hope that you get at least a sense of why we're excited. So thanks very much.
Just take one or two questions.
The fact that you use maximum entropy,
yeah,
could that mean that you find yourself at a critical point?
So, as you can imagine, we spend a lot of time worrying about, uninteresting reasons why, or rather, let's say artifactual reasons why we might be driven toward a critical point. There is an argument that, so the critical point is a place where the transformation between, the Jacobian between the expectation values that you measure experimentally and the parameters that you're adjusting is starting to become singular, right? Right? The transformation from, right, the simple one, right, from the magnetic fields, which are the parameters of the model, to the magnetizations, the derivatives is susceptibility, and critical points are places where susceptibilities diverge. So what that means is that between the things you measure and the parameters you're trying to set, there's a Jacobian, and that Jacobian is becoming, well, not infinite because the system's finite, but very, very large or very, very small, depending on how you look at it. It is possible, in principle, that that diverging behavior of the Jacobian biases us, okay? Now, I don't really believe that because I think what our experimentalist friends tell us in the physics lab is it's very hard to put a system at its critical point. So, so of course, whatever argument, if I gave you an argument that said that that divergence causes a bias, well, that was on the hypothesis that one of these parameters is the fundamental one and the other one is the one you're inferring. But it's not clear which way it is, right? So, in principle, yes, there's a possibility, but it's not really about maximum entropy. It's about the space of probability distributions. I would actually say that if this is the source of the problem, then it's interesting. It's just not interesting about biology. It's interesting about statistical inference. okay? I don't really think that's what's going on here. But in principle, I mean, your question is very well taken, and it's something that we and other people are worrying about, and some people even writing papers about. I don't think that's what's going on, but. Jerry.
Are you taking divergences as a measure of criticality? There are many examples where you have, at critical points, there are singularities. They're singular, but they're not infinite.
Absolutely. We happened to see something that's looking like a divergence of the specific heat. You're absolutely right. For example, if this were the Sherrington-Kirkpatrick model, right, there would be kinks, not singularities. And in fact, somebody once raised their hand when I was giving this talk, and they said, that must be wrong because the Sherrington-Kirkpatrick model has a kink, not a singularity. I said, well, yeah, but this is a real bunch of neurons. It's not the Sherrington-Kirkpatrick model. So yes, I mean, we're still in this. We are still in a phase of this where we're exploring, and so our attention was drawn to this one. But you're absolutely right. Of course, divergences are easier to see than the other things. So.
And indeed, there are random models where you don't have a critical point, but there's a whole region.
Yes, right. So, and it's clear. So, for instance, this argument, to the extent that this entropy equals energy result from the simplified version of the model is anything like what's going on in the real model we have here, then whatever critical surface we're on is not one of the simple ones, right? And that, of course, also delights us a bit, because that's interesting, right? We have to think about what's actually going on. Yes?
What if, so like this flocking problem, you could, I suppose, go to an engineer and say, build me a little device, and I'm going to use this as a model for birds and put them together, and I want it to flock, and he'll come back with something.
Yeah.
You take some mathematical model, and then you take its behavior and you run it through this analysis.
Okay, so if you are interested in the, if you're only interested in directional ordering, you may well find the same thing because of Goldstone's theorem. If you ask about speed fluctuations, you will almost certainly get it wrong, because the speed fluctuations also have long-range correlations. The other thing that's very interesting is that if you think not about the speeds, not about the velocities, but about the positions, this flock is very liquid, right? You don't see sharp coordination. If you measure the G of R, the positional correlations, density-density correlations, you don't see ringing, okay? It's not crystalline. Many models that purport to reproduce some aspects of flocking behavior produce crystalline flocks. And there are some crystalline flocks out there, but these starlings, which are flying over the piazza in front of the train station in Rome, for those of you who know the location, if you must choose to work on these things, take it to places. It's actually a charming story. Well, let me answer your question. So you'll get, it's easy to get some things right. And getting it all right isn't so easy. We view this as, it might be that this was one, some aspects of this aren't too hard. I think the level of quantitative detail we got, considering that all we're matching is one number measured in here, is surprising. And almost all of those models have more parameters in them. On the other hand, we view it only, I think this is to the birds as the 10 neuron analysis was to the neurons. Okay. It's a step, and you think, okay, that encourages us to keep going.
The other thing I'm trying to say is that, again, the real system does have some sort of mechanism if you're capturing something universal about the mechanism.
Right. Right. And so the question is, are we capturing something which is so universal that it's true of anything that successfully flocks, or are we capturing something which is essential about the way real birds flock? And I don't think we know the answer to that yet.
You could try.
Well, we have done, so for example, I told you that what we match is the average correlation with your first shell of neighbors. Well, how big do you think your typical first shell is? So we actually just tried the n nearest neighbor, or the k nearest neighbors. And then we asked, for these data, what value of k works best, and that is to say maximizes the probability of the data, and it's well defined. The answer is 7 plus or minus 2 or something like this. But then we went to models where we knew with how many neighbors you were interacting in the model, and asked whether we could reproduce that. And the answer was that we tend to, the number that actually emerged is about 11. And so you tend to overestimate it a little bit. And the reason is because during the course of flight, right, the birds pass each other and carry the information from having been in the neighborhood off to the next one. So your effective, because we're only discussing snapshots and not dynamics, the effective range is larger. However, we don't see any sign of what in hydrodynamics would be long time tails, in which that effective range could become infinite, which it can in some of these models.
So this temperature, I thought is a constant 1. And
the real system is that temperature equals 1. So changing temperature is just a way of exploring the parameter space.
Well, what is your definition of the temperature?
It is the direction in parameter space where all of these coupling constants scale up and down, right? Scale uniformly. I'm in a multidimensional parameter space, and I'm sitting at this point. Let me now imagine that I multiply all of my coupling constants by a constant, by lambda, or by beta, actually, right? To choose the conventional Greek letter. Well, that corresponds to being at inverse temperature beta, right? Because I'm just putting a factor in the exponential. That's all that's happening. So really, I could have drawn the picture, I could have drawn the picture as I'm in this multidimensional parameter space. I happen to be sitting here, and I want to go that way. And what I'm measuring along the way is the variance of the energy. And I would never say temperature, and I would never say specific. In fact, I would actually measure log probability, right? The variance of the log probability. And so I would never say the word energy, and I'd never say the word temperature. And maybe that would be good.
All right, I think that's enough for now.
Yeah, thanks.